Hello, my name is Shichun Gu, and I'm going to talk about the last gene of the SARS-CoV-2 genome, which is ORF10. So the whole story begins when we notice that the ORF10 sequence is highly structured and conserved and only presented in the SARS-CoV-2 genome. We analyzed over 40 million SARS-CoV-2 sequences held by GUSAID and found that actually less than 1% of those sequences had a truncated ORF10 region. The following cove glue analysis also support this finding. So is SARS-CoV-2 require this region for folding purpose, or is ORF10 actually a functional ORF? We use Wuhan D614G as the backbone and employed overlapping cDNA fragments with a yeast-based recombination system so that we can mutate the ORF10 sequence. This is the predicted structure of the ORF10 region, and we mutated both start codon to a stop codon, and these mutations were highlighted in the green boxes. It is also worth noting that after the stop codon of ORF10, there is a hypovariable region at the end, and which will be mentioned in the following slides. Although previous attempts trying to replace the whole ORF10 with either the M neon green or M scarlet reporter protein had failed, this ORF10 knockout virus was recovered successfully. Wild-type and knockout virus appeared transcriptomically similar, and when we infect host cells for 18 hours, the quantitative proteomic did not pick up any significant difference. When we mix wild-type and knockout virus in 50-50 and use them for competition assays, the wild-type virus always dominant in viral cell lines, but never dominant in the A549 cell lines. Then, thanks to our collaborators from University of Liverpool, they infected some hamsters and found that knockout virus is highly attenuated in animal models. The qRT-PCR results indicate that the wild-type replicate tenfold better comparing to the knockout virus only in lung tissues, but not in nasal or throat swabs. Then we move on to sequence those lung samples from animal models using an applicon-based nanopore sequencing. As expected, the wild-type virus is very stable. However, we also find out there is a widespread reversion of the knockout virus at both mutated AUG site. And in one of the 6 samples, the wild-type sequence actually becomes the dominant one. So what we can conclude from all those findings and what are we doing at the moment? First of all, there is no report worldwide in all the laboratories that they can detect ORF10 protein. So we are trying to find a reliable and sensitive method to detect this specific protein. Secondary, wild-type and knockout virus behave very different in hamsters models, but this difference is not significant enough in cell cultures. We suspect a specific cell line is required for ORF10 infection. So we generate a stem cell-derived human lung organelle model. Thirdly, is ORF10 knockout virus reverted back to the wild-type because the RNA structure is needed? We designed a synonymous changed ORF10 region and generated a specific ORF10 virus. So this is the table listed all the attempts we've tried to detect ORF10 protein. We have employed 3 different tags, either the 6 histone tag, the flag tag, or the triple flag tag. They were added either at the C terminal or the N terminal of the ORF10 sequence. They were introduced in either the viral system or the bacterial system. With all those different methods we've tried and listed in the headings, it is still very hard to detect this specific protein. And so far, we can only detect a shorter version of ORF10, which will fuse to the 6 histone tag when we use a bacterial system. And that would require us to use shorter versions of ORF10 amino acid as input for the mass spectrometry. This is the very long timeline scale for the induced pluripotent stem cell-derived human lung organelle model. These stem cells are treated with specific reagent so that they can differentiate in the pathway we wanted them to be. Phenotypical markers were assessed at specific time point listed in the red boxes. After 2 months of growth, we infect those LV-like structure using either the wild-type virus or the ORF10 knockout virus for 4 days. The qPCR against nucleocapsid protein expression indicate that these organelles can be infected by wild-type virus, but can't be infected by the knockout virus. Alveolar type 2 cells is the only cell that can co-sting with SARS-CoV-2 nucleocapsid protein. Then we have the stock of the ORF10 proteins which were genetically modified, and we are planning to use them for following experiment. So at first, we tried to replace the whole ORF10 region with fluorescent reporter proteins, but that failed, which might suggest the ORF10 region is needed. So we move on to the knockout virus, where we keep the RNA sequence the same, and theoretically, it cannot produce any proteins. This virus was recovered successfully and is stable during viral passaging experiment. It is infectious, but it's less fit comparing to the wild type in animal models. Now we move on to the codon synonymous changed ORF10 sequence, where we completely disrupt its RNA structure, but it should still provide the same protein. We highly suspect this virus was recovered, but it grew slower and was less infectious, although this would require following experiments to back up. We also have 2 different types of tagged ORF10 virus. The 6-cysteine tag and flag tag were carefully designed to embed it in the hypovariable region right at the end of the ORF10 sequence, so that it can have the same RNA structure and produce the same protein. Both of those tagged viruses were recovered successfully, and they are our tools for following detection of ORF10 in viral infectious conditions. In conclusion, it is still very challenging for us to detect ORF10 protein, but with all those adjusted conditions and data accumulated in the mass spectrometry, we started to see degraded peptides. And hopefully, with all those tagged viruses, we are able to pin down the cellular process in infected cells. Cycloheximide, which is an inhibitor for protein synthesis, a shorter cycloheximide treatment can lead to rapid degradation of all over-detected tagged ORF10 protein. Alveolar type 2 cell is the only cell type revealed that can be co-sting with nucleocapsid protein after our SARS-CoV-2 infection in lung organelle models. We are planning to focusing on obtaining and growing those primary AT2 cells and use them for further validation. Although sequence reversion suggested there is a very strong pressure Although the sequence reversion suggested there is an extremely strong selection pressure for the ORF10 knockout virus to reverse off in hamster studies, we still cannot rule out the importance of those RNA structures. And since the synonymous change virus is under recovery, it will be closely monitored for any sequence change in the following weeks. Finally, I would like to thank the rest of the team and our collaborators from University of Liverpool and the funding bodies. Thank you.
That was fascinating, thank you. With your reversion viruses, did you only see true reversion, or did you ever see any pseudo-reversion mutations?
Could you identify like which one is a pseudo-reversion and which one is a...
So a change that doesn't put it back to wild-type sequence, but that, for instance, might restore base pairing that you've disrupted by mutating the AUG codons?
Because we only changed 4 different bases for the knockout virus, and if it reverted back to the wild type, it is quite obvious. And I think it is real reversion.
Very early on in your talk, you mentioned that you look at GIS-8 and you saw that a very small portion of all samples had their ORF10 removed. Is that?
Yeah, so like less than 1% of all those.
I'm curious,
sequences.
when was that time that you checked? Was it pre-Omicron or very recently you looked at the GIS-8? Because my understanding is there is this premature stop codon in ORF8, and anything downstream of that in some of the major lineages, if you like, of SARS-CoV-2, which with that premature stop codon would have their tail end of their genome truncated. So I wonder if you've seen that effect. When was it?
So the first attempt when we try to search using GIS-8 is at the beginning of the pandemic, but also like we read about the literatures and found that a group in Korea, for example, they said there is around 10% of truncated sequences. So we double check after the Omicron has emerged. So it's quite recent. And the finding is still the same, less than 1%.